Yes. Then you have your trophic level 2, which are primary consumers. These are your what you call grazers, herbivores. Oh. Then you have your trophic level 3. These are your secondary consumers. They're usually carnivores, predators. Then you have your trophic level 4, which are your what? Tertiary consumers. Oh. Can do forgot how to spell tertiary. Well, let's do this. You can use this as well. So 0 above 3 means tertiary consumers, and then you can say for secondary consumers you can use this. Oops. Etc. Anyhow, the answer is secondary consumers. Let's carry on. Part C. First one. Calculate the percentage of energy transferred from the plants to the tertiary consumers. Oh, that's tertiary. That's how you write it. Yes. So percentage energy transferred to the tertiary consumers. So we're gonna go back.
I don't know how to do this calculation.
Let me do it together, don't worry. Plants is what? Plants are the trophic level 1, aren't they?
Yeah.
Okay, so you have the energy of 20,810 kilojoules. Kilojoules per meter squared per year. Okay, which is transferred to what? Tertiary consumers as what? The last trophic level. So it's gonna be trophic level 4. And kilojoules per minute there is 21. Okay. Oops. That's it. So what percentage of 21 is 20,000? So we're gonna calculate what percent is 21 of 20,810. Okay?
21 bölü 20.810 çarpı 100.
Yes. How do we do this? Do you remember the way I showed you? So in 2,810, if it's 21, what is it in 100? Okay? So you multiply these two and you divide it by wait, what's wrong with here? The 21 sayı yüzde kaçtır? So if you multiply 100.
Hocam ben de geldim.
Hoş
Geldim
geldin.
hocam. Hoş bulduk. Kaçıncı sorudayız? Bir dakikacık.
4 C.
Tamam hocam.
Yeah, I still get the same answer. So 100
Hocam page kaçtasın?
332. 300 hatta 333.
Tamam okey.
100 multiplied by 20 divided by 20,810, which gives you what, 0.1 something something 91, which in short is 0.1. %0.1%. That's your answer. Okay, does that make sense?
Let me try another way to calculate. I always
The one I did here is foolproof. Okay, you ask yourself if if in 20,000 it's 21, what it is in 100. Okay, so here what you do, you multiply these two, 21 and 100. So the ones that are parallel on sideways, okay, these two, you multiply them and you divide it by the number on here. Then you get your answer. This is ratio calculation. This is the easiest way you can do IGCSE biology calculations. You can use this in almost every question.
can do
You don't need to
21
remember.
over.
You can do the one in here as well. 21 divided by.
Yeah, I did like that.
But why? How do you remember how to do that?
Hocam.
I just know.
Direkt
Hard to say.
direkt ratio yaptınız burada.
Yes.
2000 işte 20.810'da kaçsa işte 20.810 21 ise yüzde kaçtır mantığıyla.
Exactly.
Aynen.
You can use this. You can remember this. This is not hard to remember. If you remember this, you can solve every calculation question. You don't need to remember anything else, any other formula. You need to remember this. But do you get this, Ken? Because I don't think you got this.
Yeah, I get it. I know.
So you say, for example, it's asked what percentage of 21 is of 20,000.
1%.
So how to calculate this? I'm explaining it to you again. If into to 20,000, if it's 21, okay, what it is in
Okay.
100. You put them like this. We put a question mark on the place we don't know. So these are underneath each other, aren't they? 120,000. But these are sideways. So the ones that are sideways, they are crossed together. We multiply them. We multiply 21 and 100, and then we divide it by 20,000. Then we get our answer, which is 0.1%.
Okay, I remembered.
Okay?
Yeah.
Yes, let's carry on. Second part. Explain why not all the energy contained in the plants is transferred to the tertiary consumers. So only 0.1% is transferred to the last trophic level. Why do you think this happens?
Because the organism that's eaten also respires, has movement, which loses its energy. So the rest of the energy gets transferred to the consumer.
Yes, but how?
Yani işte how.
Respiration
Uh,
is one reason for it, so you will get
Yeah.
1 mark for that.
And energy will be wasted in movement or excretion and respiration.
So yes, let's say not all the energy. If you have a complete answer, just read it to me. If you don't, let me write it. Do you have one?
Energy will be wasted in movement or excretion, and also not all the plants can be digested.
So that's a good one.
Or the part of the
cellulose.
plants, maybe more specific.
That's a nice one. Anyone else?
And they,
Okay, so
Hello?
let's say
Energy lost in process like respiration, excretion, and maintaining body temperature.
okay. So not all the energy is transferred. So none of you has mentioned that. So not all the energy G is transferred through the trophic levels. Some energy will be lost through processes, or let's say through respiration, movement, and excretion. Excretion, we don't mean feces. Furthermore, not all parts of an organism is eaten by its consumers. And some is not digested. And okay. Now all the energy is transferred to the trophic level. Some energy will be lost through respiration, movement, and excretion. Furthermore, not all parts of an organism is eaten by its consumers, and some is not digested. So you don't have a full energy transfer through trophic levels. For example, if you know grazers, rabbits, they will eat the grass, okay, but they won't eat the roots actually. They will eat just the top of it. They will not digest all of it, as you know. So rabbits, they have to go back and eat their feces, as you remember from a previous question, to digest all the glucose, all the cellulose. So thinking of it, they're not gonna go around finding all the feces, so they will lose some of the feces, which will be lost through through feces. And as you go further up from the trophic level, this energy loss will be higher and higher and higher. Okay? Because it multiplies itself. The magnitude, every time you transfer a trophic level, the magnitude increases. Then you lose more energy and more energy. Imagine, like I said, you have the plants, the grass, and the rabbit eats them. It doesn't eat all of it, it just eats the top bit. It doesn't eat the roots. Then it doesn't digest most of it, so it's digested. It goes out through feces. And you have a fox, but the fox doesn't eat all the rabbits. Okay, it only eats one, but it doesn't actually eat the hair, eat the bones. It just eats some of its muscle and fat. And then you have a loss of energy there. And then, for example, foxes, maybe they are eaten by bears or eagles. So the eagle doesn't eat all of the fox. It just eats a little bit of its meat. And it doesn't eat all of the foxes. It just hunts maybe one every week. And then because of this, you have a declining energy level as you go through trophic levels. Okay? Does that make sense?
Okay.
Yes. So part D. A student uses this apparatus to find the energy content of the plants. You have basically you're burning a sample to see how much energy is released. You have a needle, you have a thermometer, you have a boiling tube, you have a Bunsen burner, you have a plant sample. Explain how the student could use this apparatus to find energy content of the plants. Yes.
I didn't do this question. I think it's about how do you say it?
Ben
Starch test.
Nedir adı?
It's not
Yeah,
the starch test.
exactly. He should probably take the starch değilse de nedir adı? Tungsten Pulse boiling tube'a göre herhalde. Boiling tube Bunsen burner'ın üstüne şey yapar işte. Thermometre için ama Mounted Needle'da birazcık karıştırdım. Yani şu an bakınca Mounted Needle'ın ne işe yarayacağı.
Guys, it's on page 61. If you have your books next to you, it's on page 61. So it's you will put your water inside the test tube. Then you will put your thermometer inside the test tube, okay? And then you're gonna mount the plant sample on the needle. Gonna put the plant sample on the needle, and you're gonna put it on the Bunsen burner. Then you're gonna burn it, but you're gonna hold it underneath the test tube and see how much temperature changes. How much increase is there in the temperature of the water after you're burning the sample. So if you read the part, if you look at page 61, you will see what I'm talking about.
Yeah.
Measuring the energy
Can you
content
explain
of a
again,
food
Mister?
sample.
I lost my internet. I didn't hear anything.
Okay, so did you open page 61?
Yeah.
So you have there exactly the setup you're gonna use. You're gonna put your water and your thermometer in your test tube. You're gonna take your sample and you're gonna put it on the needle. Then you're gonna burn that sample underneath the test tube and see how much the temperature increases. And then you through a formula you can actually calculate the energy content of the sample. But let's read page 61 first. First of all, the mass of the food sample is found by weighting it on a balance. A measured volume of water, 20 centimeter cubes, is placed in a boiling tube, and the tube supported in a clamp on a stand as shown. The temperature of water is recorded. So first things you need. You're gonna weight your sample. How much does it weigh in grams? Second thing, you're gonna fill it up. You fill your test tube up, and with a specific amount of water, so 20 centimeter cubes, or every time you're gonna use the same volume of water if you're doing it with different samples. Okay, so that's important. And then you're gonna basically measure the initial temperature of water. If you don't measure it, you don't know how much it's gonna rise. Anyway, the food is spared on the end of a mounted needle and then held in a Bunsen burner flame until it catches fire. This might take 30 seconds or so. When the food is alight, the mounted needle is used to hold the burning food underneath the boiling tube of water, so that the flame heats up the water. This is continued, relying the food if it goes out until the food will no longer burn. So you basically keep relighting it. You relight it, you put it under the the test tube, you heat the water up. If the fire goes off, you light it up again until basically there's nothing left to light up. The final temperature of water is measured using thermometer to stir the water gently to make sure that the heat is evenly distributed. Two facts are needed to calculate energy content of the pot. The energy needed to rise the temperature of 1 gram of water by 1 centimeter cube is 4.2 joules, A volume of 1 centimeter cube of water has a mass of 1 gram. Multiplying the rise in temperature of water by the mass of the water and then by 4.2 gives the number of joules of energy transferred to the water. Dividing this by the mass of the food gives the energy per gram. Does that make sense to you guys?
Yes, yes.
Ya yani
So
şimdi
it's
anlattığınızda
really hard.
kesinlikle var.
Okay. So you need two facts. You have two assumptions here that you already know for the calculation part. The practical part, I think you got it. Did you?
Yes. Yani
50-50.
personally speaking, I did understand it.
The practical part.
Yeah.
Okay.
Yeah.
So I'm gonna go over the practical part again. In the question, it says you have a boiling tube, a mounted needle, a thermometer, a plant sample, and a Bunsen burner. Okay?
Okay.
You're gonna take your plant sample, you're gonna weight it,
İlk önce evet şeyini bulalım, masını.
then you're gonna mount it on the needle. You're gonna put it on the needle. You set that aside, you take your boiling tube. In your boiling tube, you
Water.
put a specific volume of water. Let's say 20 centimeter cubes. And then you place your
20 grams.
thermometer inside the test boiling tube. Okay? It gives you a hint here. It's a boiling tube, so you're gonna boil the water, sort of. Then you're gonna place the boiling tube on a stump, so or you're gonna hold it in your hand. But if you're usually they place it on a stand, so it sits still. Then you're gonna take your needle.
Evet.
Then you're gonna take your needle, and on the needle you have your food sample. You're gonna put it on the Bunsen burner flame. It's gonna catch fire. When it catches fire, you're gonna place it under the boiling tube. If it dies off, the fire dies off, you're gonna do this again. You're gonna catch it fire again. You're gonna put it under the boiling tube again. So until there is nothing left to burn, you do this. You continue doing this. After that, you actually measure the volume. So you stir the thermometer and then you measure the temperature of the water. So first you measured it, initial temperature, and then after you burned the food sample. So you will see an increase in the temperature. This difference in the temperature will help you calculate the energy content of the food sample by making these assumptions on the below. So it said the energy needed to rise the temperature of 1 gram of water by 1 centimeter is 4.2 joules. A volume of 1 centimeter cube of water has a mass of 1 gram. So you have 20 centimeter cubes, which means you have 20 grams of water. So if to rise the temperature of 1 gram water by 1 centimeter,
Dur.
you need 4.2 joules. So let's go back to formula. You're gonna calculate in joules because you have on the bottom, easiest one, you have the mass of your food sample. Okay, mass of your food sample is easy. We measured it, and that's it. On the top, you have final temperature minus temperature at the start. You have the difference in temperature. So how much did it increase actually? Then you multiply it by 20 grams because you have 20 centimeter cubes of water, don't you?
Yes.
Then because 1 centimeter needs 4.2 joules, you multiply it by 4.2. These are set numbers, 4.2. It's a set number. You're gonna use 4.2 every time. But the gram of
Okay, I
water...
understand.
So guys, let me write this here for you.
Şimdi ben bir şey sorabilirim peki hocam.
Yes.
Tamam. Bu formülayı bize verecek mi peki sınavda mesela atıyorum yoksa bizim kendimiz bilmemiz gerekir. Muhtemelen ezberlememiz gerekir. Yani bu formülayı.
Yes, it's not hard.
Yani aynen çok da
If it wants you to calculate it, he's gonna give you the calculation, the formula.
Oh.
But if it asks this in a question like this, you should know how to mention the formula.
You should explain. Tamam. Sure.
You should know how to explain it exactly. But you don't need to explain this because you have different points that you can get marks from.
Yani.
But in order to learn this properly, you actually need this formula.
Okay.
So first thing we said, difference in temperature. In temperature, which is what? Initial temperature.
Minus final temperature.
Sorry, final temperature. Sorry, other way around.
Final temperature.
Minus
Minus
initial.
initial. Yes, yes.
Okay. So, for example, let's say we start at room temperature and then now it's 30 degrees. Room temperature is 25. So this will be 5. Okay. Then you multiply that by, in this case, you said 20 grams. Why 20 grams? Because 1
That's how
centimeter
much,
cube of water
have.
is equals to 1 gram. If you have 20 centimeter cubes, you have 20 grams. If you have 10 centimeter cubes, you have 10 grams.
Right.
Then you have your set number 4.2. 4.2 to basically it is the energy needed to heat up 1 gram of water by 1 degrees. Energy needed to heat up 1 gram water by 1 degrees centigrade. Okay, does that make sense to you?
Yeah.
Because we have 20 grams, you multiply it by 4.2. If we had 1 gram of water, then it's just you multiply. Well, it doesn't change anything. The last thing we do, we divide it by mass of the food sample.
O da bu durumda ne kadar
In-
olur?
Grams.
Görebilirim.
It doesn't matter. Depends on your mass of the food sample.
Heh, yani.
It depends how much does it weigh.
Exactly.
You want an let's do an example. So you have a difference in temperature. We said 30 degrees minus 25, which is 5. Okay. Then 5 is multiplied by 20 grams. Let's put it in brackets. Then you have 4.2, which is divided by, let's say, how many grams it should be? Say 20 grams. This is your mass. So let's calculate this. 5 multiplied by 20 multiplied by 4.2. Then you divide it by 20. You get 21 joules, for example.
Ah.
To make this kilojoules, you have to
Multiply it by 100, 1000 sounds.
divide it by 1000. Because kilojoules.
Kilojoule.
But
Multiply it.
state it like this. Because this is not a calculation question. This is just an example for you. This is not the actual answer for the question, okay? So by knowing all these, you should be able to write an answer to this question. Am I right?
I didn't
For
understand.
sure,
Like,
kesinlikle.
is the 20 multiplied by 20 grams? Is this always like?
What do you mean? I don't understand.
Okay. I'm stupid. Okay, I understand. I see.
20 grams is there because you use 20 centimeter cubes of water. Each centimeter cube
Yeah,
of
I
water is
understand.
1 gram.
Okay.
Just like 1 liter of water is 1 kilo, approximately. Same
Okay.
assumption. So let's write an answer to this. So question says you have this apparatus. Explain how the student could use this apparatus to find the energy content of the plants. Firstly, the mass of the food of the plant samples be calculated or measured in grams. Okay.
Şey desek olur mu?
After that,
Weight in grams desek olur mu?
yes, you can say that.
Should be.
After that, the test tubes should be filled up with a measured volume of water, 10 centimeter cubes, with the thermometer placed in and secured at a stand. Okay, that's it. The apparatus is ready. Using the mounted needle, the plant sample will be allowed to catch fire on the Bunsen burner. After it's alight, it will be placed underneath the test, not the test tube, boiling tube. If the fire goes off, it should be repeated until they're left to burn. Test tube, where did I say test tube again? Okay, and the increase in the temperature of water. Oh, we forgot to mention they should measure the water. The increase in the temperature of water should be calculated after the Yeah. The increase in temperature of the water should be calculated after burning the sample. Then you could use by multiplying the change in temperature with the mass of the water inside the boiling tube and 4.2
So do we write the equation into the question also?
not if you want, but not necessarily. So by multiplying the change in temperature with the mass of water inside the boiling tube and 0.2, thus dividing it by the mass of the sample, plant sample, energy in joules can be calculated. Okay. The process can be repeated with other samples to take an average energy content for accuracy and reliability. Yes. Let's read this again. Firstly, the mass of the plant samples should be measured in grams. After that, the boiling tube should be filled up with a measured volume of water, about 20 or 10 centimeter cubes. Why do we do this? We do this in order to calculate the grams of water. Easier. With thermometer placed in and secured at a stand. Initial temperature of water should be measured. Using the mounted needle, the plant sample will be allowed to catch fire on the Bunsen burner. Let's say Bunsen burner flame. After it is light, it will be placed underneath the boiling tube with water. If the fire goes off, this should be repeated until there is nothing left to burn. The increase in the temperature of water should be calculated after burning the sample. By multiplying the change in temperature with the mass of the water inside the boiling tube and 4.2, thus dividing it by the mass of the plant sample, energy in joules can be calculated. The process can be repeated with other samples to take an average energy content for accuracy and reliability. Other, let's say, plant samples. Yes. Does it make sense for everyone?
Yeah.
Okay. Let's carry on.
It makes sense.
Next question. A student investigates how light affects photosynthesis by looking at changes in carbon dioxide levels. The student uses this method. Step 1. Place 2 cm cubes of orange hydrogen carbon indicator solution into each of 3 test tubes, A, B, and C. Put a leaf in tube A and leaf in tube B. Wrap tube B in aluminium foil. Seal all 3 tubes with bungs. Place the tubes in a water bath for 2 hours. Explain the purpose of tube C. Yes, guys.
Compare with the tube A and B.
Why? Because there's nothing in there. So what do we call an empty test tube usually.
Onu
See-
düşünüyorum şimdi 2 saat.
Control?
Ama asla.
Yes, exactly. Control.
Galiba
Its only purpose is control. To see if any effect is actually caused by the leaf, not just the air. Does that make sense?
Yes.
yap.
So if you have no leaf, you can see maybe the color is gonna change on its own. You don't know. So it's to control.
Kontrol istiyorum.
Hey. Okay. Yeah, that's probably a full answer. Anything else you guys want to add?
No.
Yani sizinki gibi yazdım ben de.
Efendim?
Ben de sizinki gibi yazdım işte. For control to see if anything is caused by the leaf.
Exactly. Second part, why it is necessary to measure the temperature of the water bath. So, students investigate how light affects photosynthesis. Temperature is a factor that affects photosynthesis, isn't it?
Yes. And light intensity.
Exactly. Temperature is another factor that affects.
Can I say to keep the temperature constant?
They're not doing anything to keep it constant, really. It's just measuring it.
Okay.
They're not saying us that they're keeping it constant. By measuring the temperature, they are checking if it stays constant, basically. Okay. Any other answers?
Sadece constant water temperature desem olur mu hocam?
Not really. Constant. They don't keep it constant. They don't say any. The water bath, it doesn't have a temperature. It doesn't say. They don't say us. They're keeping it constant. They just say they're measuring it. And the question actually asks why it is necessary. Why is it necessary to measure temperature of the water bath? To see if it changes, basically, because temperature affects photosynthesis. Does that make sense?
Yes.
No. Carrying on. Name the independent variable in this investigation.
Light durumda.
Light. I also write light.
Light intensity.
Yeah.
Because
Light aynen.
yes, so it doesn't depend on a thing because photosynthesis depends on the light, and the thing you are basically using, the thing you are changing, is your independent variable because you're controlling it. It is not being affected by anything else. You're changing that to see what effect it has on other stuff all the time. So thing you are controlling is your independent variable. Okay? So almost all of the time, because you change something to see its effect on something else. So it won't depend on anything else. It will only depend on your opinion, choice, for example. So it won't depend on any other variables. You change it as you please to see its effect on other variables. So light, yes.
Hocam en son ne anlattınız siz?
En son derken?
Yani en son şeyden bahsederdiniz nedir adı? Light intensity'den. Sonra başka bir şeyden bahsettiniz mi?
Anlamadım. Nasıl yani?
İnternet.
İnternet gitti.
İnternetin
Ha.
gitti geldi daha.
Sadece I said yani
Tamam.
independent variable is the variable that you control.
Tamam.
Because it doesn't depend on anything
Tamam.
else. It depends. It
Evet.
does. Some other stuff depends on it. Rate of photosynthesis depends on the light intensity, and you are controlling light intensity. So your independent variable is light, or light intensity.
Tamam.
Okay. The table shows the students' results. Explain the change in color of the indicator in the test tube A. So test tube A, you had light, as you remember, and you had hydrogen carbonate solution. So hydrogen carbonate, it changes color. When there is carbon dioxide, it is orange. When there is not, it changes color.
Okay.
So as it increases, if the carbon dioxide level increases, it turns yellow. If it decreases, it becomes more red. So in test tube A, as you see, because there was light, carbon dioxide,
Photosynthesis.
yes, and then the carbon dioxide was being used. So after 2 hours, it became red. In test tube B, it was dark. There was no photosynthesis, but there was what?
Starch is up.
So what is that?
I forgot the respiration, yeah?
Respiration. So there was a production of carbon dioxide. Because of this, it turned yellow. In C, nothing happened. It stayed orange. Okay? Explain the change in color of the indicators in tube A. Yes. Any answers?
Photosynthesis.
So there was light. Thus photosynthesis was able to take place using up the CO2. That's it. Or you can say there is less CO2 because the plant is photosynthesizing. The leaf is photosynthesizing. Now, second part is what?
Use up the starch respiration.
Explain the change in color of the indicator tube B. So it become yellow. So the leaf wasn't able to photosynthesize. Without light, but was respiring and releasing CO2, increasing its concentration. Okay?
Okay.
But C suggests a different methods that the student could use to show that light affects photosynthesis in leaves.
Hocam ben düştüm. Bir daha anlatabilirsiniz.
Of course.
Şey
Nerede kaldın?
en son işte i'nin cevabını açıklamadıydınız daha. Direkt karbon bahsederdiniz. Nedir adı? İçindeki solution'dan. Hydrogen carbonate.
So part 1 explain the change in the color of the indicator in tube A. So because there was light, the leaf was photosynthesizing, so it was using up the carbon dioxide. Okay?
Okay.
So we wrote it down here.
Tamam.
Second part, the leaf wasn't able to photosynthesize explain the change in color of the indicator in tube B. The answer is the leaf wasn't able to photosynthesize without light, but was respiring and releasing CO2, increasing its concentration in the tube. Because it wasn't using up oxygen. Oh, sorry, no oxygen, carbon dioxide. It was producing more CO2.
Okay.
Part C suggests a different method that the student could use to show that light affects photosynthesis in leaves. So now you're gonna mention about starch if you want, or you can mention about the experiment we did. Remember the experiment we did with the Elodia?
Yeah. The bubbles you can see.
The bubbles. O zaman yapabilir. The student could cut a piece of underwater plant and put it inside a small container, and should place the light approximately first 5 centimeters away from the light source.
We didn't cut a leaf actually. We cut a section of a plant.
Evet. Tam olarak leaf değil.
Elodia, Canadian pondweed.
Elodia. Evet.
You start again.
Okay. So a section of the Elodia plant is used in this experiment, and a light source is placed in front of it. First, 5 centimeters away. Yani öylesine söylüyorum şu an. Kaç centimeter olmasına gerek yok. And then first 5 centimeters, then 10, and then 15. For control, another plant can be used for more accurate results. Ve böylece de yapabilir. Nedir adı? And then the student would obtain the result and calculate how much photosynthesis occurs in the leaf.
So you can how can you, because you didn't measure anything, you didn't mention that you measured anything. So you're gonna mention that you got measuring bubbles per minute. That's how we measured the rate of photosynthesis in that experiment.
Distance from light.
Sorry?
Distance from light.
Like distance from light is measured via a ruler or about in centimeters, and then you measure the rate of photosynthesis by counting the bubbles per minute. So let's write it down. Give me a second. Okay. The student can use the aquatic plant Elodea and measure the rate of photosynthesis at different light intensities. A section of the plant Elodea can be used upside down in a test tube filled with water. The test tube can be placed at different distances from a light source, increasing or decreasing light intensity. Okay. At each each distance, rates of photosynthesis can be calculated by counting oxygen bubbles produced per minute. Okay. The student can use aquatic plant radar and measure the rate of photosynthesis at different light intensities. A section sorry. A section of the
Çok
plant
yaşa.
I didn't sneeze, Ken. Anyhow,
You did, I thought.
I just coughed. Okay. Anyway, a section of the plant radar can be used upside
Allah.
down in a test tube filled with water. Believe me, I'm not Corona. Okay.
No.
Okay.
The test tube can be placed at different distances from a light source, increasing or decreasing light intensity. At each distance, rate of photosynthesis can be calculated by counting oxygen bubbles produced per minute.
Okay.
I mean, this should be enough.
Ya. Yani de şeyden bak. Experimental.
Is that clear for you?
Yeah.
Yes.
Yes.
I choose this one. You can do starch as well, but I chose this one because it's an experiment with it. And we have our last question. Question 6. Diagram shows section through the skin. Name the structures labeled A, B, and C.
Bunu yapıyorduk galiba.
Aa, bunu bir yerde yaptık doğru. Ama sanırım mock'ta soruyorum. Kim soruyor? Yoksa
It might be an
bir yerde
exam question, I think.
sorduydunuz. Sanırım exam question.
Anyhow. So what's A, B, and C?
Aslında bu diyagramı ezberleyemedim ben hiç. O yüzden hiçbir fikrim yok.
So, obvious one is C, which is hair. Okay.
Aynen.
Here and B is capillary. A is sweat
B
gland.
is blood vessels.
Kapiller izlesek olur mu peki?
Can I say capillary?
Or capillaries, you can say that. Capillaries are blood vessels anyhow, so. And A is the sweat glands. Okay, or you can say ducts. Or sweat duct. That's clear. The last part. Explain the changes that occur in structures B and C when a person responds to being in a cold environment.
Şimdi bunu anlatması aslında güzeldir, çok güzel. Da şu an yazamam.
Let me check.
Hiçbir şey yapmadım.
I think I did. Yeah, I did it. The capillary become narrower and the blood flow to the surface of skin reduce the wait, can I read again?
Yeah, vasodilation, for example.
The capillary become narrower and blood flow to the surface of the skin.
Reduces.
So less heat is lost in this case. It's called wait, how do I say that word?
Vasoconstriction.
Vasoconstriction.
Vasoconstriction.
Construction bölümünden.
That's it?
Yeah. I think I
Anyone
get
else?
maybe
I could write a great answer ama it would take me like a few minutes. O yüzden
We should
direkt
have done it
yazdım
before though.
istiyorsan. Exactly ama yapmadım.
Guys, don't be lazy. Come on, do these questions. Um, this is an actually easy question. Let's think first. So you have it actually gives you the structures as well. So you know what the structures are gonna do at some changes. A is not gonna do anything because A acts when we are hot. We don't sweat when we are cold, do we?
No.
So what happens when we are cold? So our hairs
the
they
blood.
stand up. Okay, the first
Yes,
thing, our
to
hairs
keep
they
the
stand
heat.
up. It doesn't work for
Exactly.
us actually.
To prevent.
It works for other animals because we don't have enough hair. For example, it
Aw.
works for birds. It works for small mammals. It doesn't work for us usually,
Evet.
but it happens. Blood vessels. So we get paler when we are cold. This is because we have much less blood flowing closer to the surface. So the capillaries they constrict, they carry out vasoconstriction, they get narrower, uh, reducing the blood flow to the surface of the skin. This makes heat loss through radiation. It increases heat loss through radiation, and that's all of it really. There's nothing else you can mention. So let's write a simple answer. Part B, isn't it? Yes. Oh, what is this?
Hocam ben de okuyayım cevabı yoksa.
Ah, yes. I didn't know you had an answer.
What?
Oh.
Vasoconstriction occurs which reduces blood flow to skin.
Mhm.
Hair close to skin prevent heat loss.
Mhm.
Also, adrenaline causes increase in metabolism, then shivering takes place, which produces heat with the relax and contraction of muscles rapidly, and the person will look paler.
Okay, so the wrong thing with your answer, the last part is not necessary, because if you read the question, explain the changes that occur in structures B and C. Okay? Do they do that, B and C? No, they don't have anything to do with adrenaline. But it's correct. I mean, it's not wrong, but it's not it doesn't fit the question. Anlatabildim? Yani question onu sormaz sana. B ile C'yi sorar sadece. Yanlış değil ama question onu sormuyor. So capillaries near to the surface constrict, decreasing the blood flow nearer to the skin surface. This process is called vasoconstriction and prevents most of the heat lost by radiation through the skin. Although less apparent in humans, in cold hair stands up by the construction of hair erector muscles. These traps air around the skin, creating insulation, preventing heat loss further. By, let's say, convection. Yes. Capillaries near to the surface constrict, decreasing the blood flow nearer to the skin surface. This process is called vasoconstriction and prevents most of the heat lost by radiation through the skin. Although less apparent in humans, in cold hair stands up by the constant contraction of the hair erector muscles. This traps air around the skin, creating insulation, preventing heat loss by convection further. Okay, that's it. Is that clear?
I basically write the same, but I think just some detail.
So you want to mention about vasoconstriction. You're gonna mention narrowed, less blood that flows towards the skin surface. The hair erects by the contraction of the muscles, and it insulates air. And all this, they prevent heat loss by convection or radiation. And we actually mentioned all of these in our sentence. We only need to mention 5 of them to get 5 marks, but we mentioned 6 of them just to be just in case. Yes, guys, this paper is finished as well.
Next class'ın herhangi bir paper falan bitirmemiz gerekir mi?
I think we finished all of them, didn't we?
Bilmem. Do we?
We didn't finish all of the past papers, I think.
Which one we didn't finish?
Check, I don't know.
Şey yapılı değil ama bir kısmı yapılmış olabilir.
Let me finish the recording first.
Yeah.